Welcome to this presentation on data collection and analysis in healthcare-associated infections, HAI. Today we will explore the systematic process of turning raw clinical data into actionable insights for the Hospital Infection Control Committee, HICC. Slide 2, the Surveillance Lexicon. Before we dive in, we must align on our terminology. HAI refers to infections monitored to reduce hospital risk, primarily CAUTI, CLABSI, VAE, and SSI. The infection control nurse, ICN, is the backbone of this process, performing active, laboratory-based ward liaison surveillance. The HICC is the multidisciplinary body that analyzes this data, while the NHSN, National Healthcare Safety Network, provides the gold standard criteria we follow. Slide 3. The continuous framework of HAI surveillance. Surveillance is a continuous cycle, not a one-time event. It begins with data collection via daily visits and clinical correlation. Next is data analysis, where we generate monthly location-wise rates. We then move to data interpretation, applying risk-adjusted metrics like DUR and SIR. Finally, we ensure data dissemination through monthly reports to the HICC and clinical departments. Slide 4. Stage 1. Active surveillance methodology. In Stage 1, we employ a targeted strategy. We focus on high-risk locations like ICUs and transplant units using the laboratory-based ward liaison method. The ICN workflow involves daily visits to check patients on devices and post-operative cases, cross-referencing clinical signs with microbiology laboratory investigations before entering data into standardized forms. Slide 5. Capturing the numerator. Form 2 is our tool for documenting the numerator, the infected patients. It captures patient demographics, the presence of devices, and daily clinical signs like fever or purulent drainage. Crucially, it includes a microbiology culture report with the organism isolated and its susceptibility profile. Remember, this form tells us who is infected, but it cannot determine the rate of infection on its own. Slide 6. The importance of the denominator, Forms 4 and 5. To find the true incidence, we need the population at risk, the denominator. Form 4 tracks occupied beds and device days in adult pediatric units. Form 5 is used for the neonatal ICU, where risk is stratified by birth weight categories, as smaller infants face significantly higher risks. Without exposure data, infection counts are epidemiologically meaningless. Slide 7. Calculating risk exposure. In analysis, we distinguish between patient days and device days. Patient days measure the total days all patients are admitted to a location, providing a sense of general occupancy. Device days measure the total days patients are exposed to specific invasive devices, like central lines or ventilators. In short, patient days measure presence, while device days measure specific risk. Slide 8. Stage 2. Data analysis and rate standardization. Stage 2 involves converting absolute infection counts into standardized location-wise rates. Following NHSN standards, we divide the numerator by the denominator to generate monthly reports. This allows for 3 critical comparisons: tracking a location's performance over time, comparing different units within the same month, and benchmarking against national NHSN percentiles. Slide 9. Device-associated infection DAI rates. For DAIs like COUTI, CLABSI, and VAE, the formula is number of cases divided by number of device days times 1,000. Using a multiplier of strong 1,000 device days standardizes the incidence, allowing for an apples-to-apples comparison, regardless of whether a unit is large or small. Slide 10. Procedure-associated infection rate. Surgical site infection, SSI rates, are calculated differently. Unlike devices with ongoing exposure, surgeries are discrete, single events. Therefore, the strong SSI rate is calculated as strong number of cases divided by number of surgeries done, times 100. This expresses the prevalence as a percentage of operations performed. Slide 11. Stage 3. The limitations of raw rates. Raw rates can be a trap. Comparing a facility that performs primarily clean elective surgeries, Hospital A, to one managing dirty trauma surgeries, Hospital B, is unfair without adjustment. To account for patient population complexity and baseline risk, we must move beyond raw numbers to risk-adjusted metrics, like DUR and SIR. Slide 12. Assessing vulnerability. Device Utilization Ratio, DUR. The DUR measures the proportion of patient days during which a specific device is used. The formula is strong number of device days divided by number of patient days. A higher DUR indicates a higher inherent risk of an HAI occurring in that unit, independent of the quality of infection control practices. Slide 13. Benchmarking performance. Standardized Infection Ratio, SIR. The strong SIR is a superior indicator for comparison across facilities. It is calculated as strong observed HAIs from Form 2, divided by predicted HAIs. The predicted value is based on national NHSN baseline data, adjusted for the specific facility's risk factors. Slide 14. Interpreting the SIR dashboard. Interpreting the SIR is straightforward. An SIR 1.0 means fewer infections were observed than predicted. The facility is outperforming expectations. An SIR equal to 1.0 means the facility is performing exactly as expected. An SIR greater than 1.0 is a red flag, indicating more infections than predicted and requiring immediate HICC intervention. Slide 15. Stage 4 and beyond. Automated surveillance. Finally, we look toward automated surveillance. By integrating laboratory information systems and clinical information systems, automated software can track HAIs in real time. This prevents manual calculation errors, enables faster dissemination, and provides automatic alerts to clinicians. Automation allows us to transition from being mere data collectors to being data action takers.